Hey everyone, welcome back. Over the last few videos, we spent some time in the command line learning how industry tools process next generation sequencing data. But as you might know, our course name is Bioinformatics with Python. So we are now moving back to our roots. We will not be focusing this much on command line tools. Actually, we will be focusing more on Python. Now today we are going to solve one of the most annoying problems in bioinformatics, which is manually downloading files. So like if you need 50 different gene sequences, clicking download FASTA 50 times on the this NCBI website is a waste of your time. So today I'm going to show you how to use BioPython's Entrez module to write a script that talks directly to the NCBI servers and pulls the DNA straight into your Python variables. So let's jump into VS Code. So let's set up Entrez first of all. Entrez is a research and retrieval system used by the NCBI, which houses GenBank, PubMed, and more. So BioPython has a built-in module that lets us interact with it using an API. So let's import it. There is one golden rule you must follow. NCBI requires you to tell them who you are. Okay, so if you don't provide your email, they will block your script. So first of all, we will do the importing. Okay, from bio import Entrez, and one more we will copy, and we will also import seqio. Okay, next thing we have to do is provide our email. So Entrez the email is equal to, okay, uh, and now that should do it. Now we will, let's say we want to find the DNA sequence for a specific gene. So let's look for the EPSPS gene in Oryza sativa, basically Asian rice, which is a heavily studied gene in agricultural genomics. So first we need to ask NCBI to search their nucleotide database and give us the ID number for that gene. Okay, so what we will do is we will first let's print a statement for helping us understand. Searching NCBI. Okay, this should a simple command. After that, we don't need F over here. This command search handle is equal to Entrez dot esearch database name nucleotide. Then term will be uh, Oza sativa EPSPS. And one more thing, retmax. I think that's the right command. We'll check. Okay. Then what we will do is read the results into a Python dictionary. So search results is equal to Entrez dot read search handle, and finally search handle dot close. Then we will extract the sequence ID. Sequence ID is equal to search results, and what we will do here is ID list 0, and then finally we will print count sequence ID to be what it will be, sequence ID, right? Okay, we'll add one more for the rice EPSPS gene. Okay, now let's run this command. Okay, so when we run this, we don't get the DNA yet. We just get the NCBI accession ID, as you can see here. Okay, It's like asking a librarian for a book and they give you the shelf number. So now we have to actually go fetch that book. So to get the actual sequence, we use entrez.efetch. We pass it the ID we just found and we explicitly tell it we want the data formatted as a FASTA file. Okay, so we will add a print statement first of all, fetching FASTA directly into Python. Then we will run fetch handle is equal to Entrez dot e fetch that's the command. Then database is equal to uh, nucleotide. Then id is equal to sequence id. Then read type is equal to FASTA. One more which is read mode is equal to text this time. Okay, then we will read the data using sequence IO just like a local file. So what we will do is a record is equal to sequio dot read. What we will read is fetch handle and the file format will be FASTA, right? And then finally fetch handle dot close. And we will just add some simple print statements like download complete. What else can we add? We can add print uh, header will be uh, record dot description. Similarly, we can add length and we can also print first 50 bases like this. Okay, and let's run this and see what happens. We'll wait for the email to fetch our dataset. Okay, and look at that output. We successfully grabbed the FASTA header, the sequence length, and the DNA itself. Notice that we never saved a file to our hard drive. The DNA went directly from the NCBI servers in Maryland straight into our Python record variable. So of course, if you do want to save it, you don't have to ping the server again tomorrow. You can write it to a file with one line of code using sequence icqio.write. Okay, so what we will do is with open, what is the name of the file? Rice psg.pasta. That's what we will call it. Okay, and then write mode as out file. Okay, and finally sequence IO dot write record into out file and the format is pasta. Let's just print one statement and what we will say it save to rice SPS scene dot pasta. Let's run it and here we have our pasta file downloaded as you can see. And that is how you automate data retrieval in bioinformatics. By putting this logic into a for loop, you could give Python a list of 100 or 1000 gene names and walk away to get a coffee and come back to find all 100 FASTA files neatly downloaded and processed. Python is all about making your life easier, basically automation. So in our next video, we are going to continue this flow and the script is on GitHub. So grab it and try searching for your favorite gene. Hit the like button, subscribe, and I'll see you in the next video.